Welcome to today's episode where we dive into the molecular mechanisms that safeguard genome integrity. Our guest, Dr. Mo Motamedi from the Center of Cancer Research at Massachusetts General Hospital, investigates how chromatin-based pathways regulate DNA repair, gene silencing, and genome stability. His work combines genetics, biochemistry, and high-resolution genomics to uncover how non-coding RNAs and chromatin-modifying complexes coordinate cellular responses to stress. Join us as we explore how these fundamental processes, when disrupted, contribute to cancer development and progression.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Today I'm joined by Dr. Mo Motamedi. Thank you, Mo, for joining me today. How are you doing?
I'm doing well. Thank you. Thank you for having me.
So, to ease into the interview and to warm up a little bit, I have an introductory question that I always like to ask every guest to start off the podcast. How did you become interested in biology in the first place and then in pursuing a career in science?
Yeah, so actually the story goes back to when I was younger. So my brother and I, he's older than me. I've always been good at science and math and things like that. And, the reason I went down the biology pathway was because my brother already went into math and physics, even though math and physics were always my strongest grades. And that was where my strength was. I wanted to distinguish myself from him. So I chose biology, which, by the way, was my weakest science. So chemistry was the second strongest. So I like the challenge. But, once I got into biology, I really fell in love with it, especially when I took my very first genetics class and I realized how quantitative biology can be, even though there was only a little bit of quantification that was happening at the time. But then, as we can see, the spread of quantitative biology has really taken over now. And with these large data sets, I find it quite gratifying to have some quantitative skills left in my tank.
So, since you mentioned it, I want to divert from my script a little bit. So, how did you experience this? I mean, there was first qPCR probably, or even PCR, but I mean, that's not quantitative because it's not in the name. So there was qPCR, now sequencing and all these methods currently. So how would you describe this path and how did you experience that?
Yeah, from the genetics perspective, I was very interested. I was interested in the quantitative predictions of how traits can be transmitted through generation. So that's what was the hook for me in terms of the quantitative part of genetics. And then, of course, I always thought about and wondered when we're going to have these large enough data sets where we could essentially create mathematical models of various processes and be able to make predictions on that. And now I think we're getting to that sort of stage where, some pathways and, specific processes are known well enough that we could build, I would argue, still fairly crude mathematical models, but nevertheless, they are informative in being able to make specific predictions that then could be tested experimentally. I think ultimately, in order for us to get there, let's say the ultimate goal of being a cyber cell, a cell that can be sort of created by mathematical models, in order for us to get there, we still need a lot more information and a lot more experiments. But eventually we'll get there. And I think once we get there, the speed of discovery is going to go up astronomically.
So I was inferring that, of course, it would be genetic information. But I guess when listening to you, you are not only talking about genetic information, right?
Yeah, no, it's not just genetic. I'm also very much interested in just biology, right? I mean, you can't get away. If you're doing genetics, you have to do biology. And if you're doing biology, you end up in genetics. So I think, they're all interlinked. So I don't try to silo myself into genetics or biochemistry or cell biology or all these disciplines. I think the key thing is that they all have a lot to offer. And with the overall arching goal of essentially understanding a cell. And I would argue that one of the most important things that I've learned over the last little while is evolution and how understanding evolution is so profoundly important in understanding mechanisms. And I came across that because we were doing this sort of a foray into genome evolution, and I went to a couple of these evolution meetings, and it was just so eye-opening to see all these different unique organisms that exist around our planet and how their biology is really informative about some of the things that we're interested in. So I think by looking at how organisms and species evolve, we can learn a lot, and how evolution has, of a specific pathway, for example, has diverted in these different organisms. We can learn about the mechanisms of that specific pathway in whatever that
Mm.
we're studying.
It's very interesting that you mentioned that, because it seems that I planned for all this, but it's really by chance that the 3 episodes now mention like genome evolution and enhanced evolution. So 2 episodes ago, I talked to Arnaud Pedros from Spain, and last episode is with Emily Wong from Australia. They all like have to or work on evolution, genome evolution, and it's very interesting. So I did not plan for that to be like 3 episodes in a row. But anyway, focusing more on you. So for those unfamiliar, could you introduce yourself and your current role?
Sure. My name is Mo Motamedi. I'm a PhD, and I work at the MGH Cancer Center, which was recently has been named. So it's called the Krantz Family Center for Cancer Research. It's a mouthful, but nevertheless, we're happy that the Krantz family actually endowed our cancer center, and I've been in my role for several years, and my lab is primarily focused on how cells establish epigenetic states, how they maintain them, and how they pass it on through cell division. And I'm also interested in a little bit in genome evolution and how organization of the genome tells a biological story. And so that's another area that we've been focusing on. And also, because I'm in the cancer center, I'm very much interested in cancer epigenetics, specifically how cancer cells co-opt specific machinery to establish resistance in response to long-term treatment. That's another area that we're working on right now.
So when we go back to the beginning of your research career, and maybe continuing in the origin story, you worked with Danish Moazet, I hope I pronounced this correctly, on RNAi complexes and heterochromatin protein 1. So how did you choose or get started working in Danish's lab?
Yeah, so I was a postdoc in Danish's lab. So I did my PhD in double-strand break repair, looking at how actually using E. coli as a model to look at direct PCD-mediated recombination. And after that, considering obviously E. coli doesn't have really chromatin, as we like to call it, I always wondered how, different kinds of DNA dynamics impact, important activities. For example, transcription and compaction of chromatin. And essentially, I think the thing that drew me to Danish's lab was I was very interested in understanding how a heterochromatin occludes recombination machinery from accessing repetitive DNA elements. And I still think the primary role of heterochromatin, although this could be a little bit controversial, the primary role of heterochromatin is to maintain genomic stability through repressing recombination. And I think the transcription sort of repression is a bystander side effect, which I'm not saying that it's not without function. It certainly has function, but I think it's a secondary role. The primary role is to prevent recombination. And how heterochromatin really represses recombination is still something that I would argue it's fairly wide open. And I'm not working on it, by the way.
Okay.
Interestingly.
So if you focus on the RNAi complexes first, can you talk about the work that you did there?
Yeah, so when I came into Danish's lab, it was at the time when RNAi was recently discovered and received the Nobel Prize. And more importantly, it was primarily thought to be a cytoplasmic event, right? So most of what was known about RNAi was in the context of its ability to repress translation, or let's say, disrupt RNA stability. But all that activity was cytoplasmic. And, the whole foray into how nuclear RNAi could operate, which was very unique and sort of exciting at the time, came from the discovery that in fission E cells, if you knocked out RNAi components, you lost silencing at heterochromatic regions. And so this all of a sudden put RNAi in the nucleus. And so the whole question was, what is nuclear RNAi? What is it doing? What are its functions? And now, of course, we know a lot more than that. But that was sort of like the moment that I came in. And I think the nuclear RNAi was really taking off at the time. And I was lucky to work on several really exciting projects with some really talented people as collaborators in Danish's lab, and we were able to, contribute to our understanding of how this mechanism works.
Can you summarize some of the results?
Yeah, of course. So I think the thing that really, I think the most important paper came out in 2004 in Science. It wasn't my paper. It was Andre Verdel, who was the first author on that, where when they purified the argonaute protein, which is one of the key mediators of RNAi, from Pombe, they discovered that it physically interacts with the chromatin binding protein. And that chromatin binding protein specifically bound to trimethylated lysine 9 of histone H3. So now all of a sudden you have argonaute right at chromatin. You could chip it to chromatin, and it's at heterochromatic regions. So all of a sudden RNAi came there. And now the question was, well, how does the whole mechanism work and operate? And that's where I came in with my project, again in collaboration with Andre and Serafin. We purified the RNA-dependent RNA, and Danish too, by the way. Danish was doing some of the experiments for this paper too. So we purified the RNA-dependent RNA polymerase complex, and we found that it physically interacts with the argonaute protein. And again, so the whole kind, remember that argonaute was binding to chromatin through another moiety. And so now all of a sudden you have the RNA amplification machinery bound to that complex that I was telling you about. It's called the Ritz complex, the argonaute-bearing complex. And all of that is on chromatin, assembled on chromatin. And then Dicer comes in and chops up these double-stranded RNA that are generated by RDRC, generating these sort of more siRNAs that can then recruit all the other factors. So this led us to propose this model, which still stands the test of time. It's called the nascent transcript model, according to which long non-coding RNAs that are tethered at chromatin act as assembly. They are a platform upon which all these other factors assemble. And through iterative cycles of siRNA amplification, more Ritz complexes get recruited. So that's how we were able to place RNAi at heterochromatin. And of course, because K9 methylation is there, the heterochromatin machinery is there too. So then, over the years, it was shown that Ritz complex physically interacts with the histone methyltransferase complex called CLAR4, which, the CLAR4 is the main catalytic enzyme. The complex is called CLARC or CLRC. And that complex physically interacts with Ritz. So now Ritz is able to recruit the methyltransferase, and you have more RNAi. So now this created two amplification loops. You have the siRNA amplification loop that's occurring there with all these complexes being at chromatin. And then CLAR4 comes in through its interaction with Ritz. And what CLAR4 has, it can not only methylate K9, but it can also bind to it. And again, so now this also provides sort of an understanding of how these K9 methylation can spread through this mechanism. So nucleation followed by spreading is the sort of the core concept that emerged from all that.
Another protein you were working on in your postdoc was HP1, because that's also associated to heterochromatin. And there are two homologs in this family that are required for heterochromatic gene silencing. So how do both proteins work? Was this then like a follow-up on all these RNAi, because it's also heterochromatin?
Yeah, for sure. Yeah, so the whole thing was that, at the time, in terms of what these two HP1, heterochromatin protein 1, or HP1 proteins, how they were contributing to silencing was not known. And so we knocked them out, and we looked to see, together, individually, et cetera, and we looked at silencing everywhere. And the major conclusion that came out of that paper is that the two proteins one of them is called CHP2, the other one is called SWI6. And what we found was that CHP2 is primarily working with a histone deacetylase complex, and it's involved in deacetylating K14, at least we were able to show. And it was also shown at the time. And that is really involved in repressing transcription by preventing RNA pol2 from transcribing. So that complex is really working by essentially reducing transcription at heterochromatin. Best way to say it.
So more like a deactivation than an active repression.
Yes, exactly. So that's classically known as transcriptional gene silencing. Then we found that SWI6 is acting as post-transcriptional gene silencing. So it really allows for the degradation of the transcripts after they're made. And we were able to show that these two pathways are genetically separable. So if you make mutation in one, you lose some silencing. If you mutate the other one, you lose some silencing. And you get the full de-repression when you knock out both proteins. So that then placed both transcriptional gene silencing and post-transcriptional gene silencing at heterochromatin, both of which are really required for mediating and establishing the full silent state at heterochromatin.
But they are like both part of the same complex.
They are not both part of the same complex. They form distinct complexes.
Okay.
Yeah. So that's, yeah. So SWI6 is sort of touching and working together with the RNAi machinery and RNA degradation machinery, which goes beyond RNAi. There's also exosome-mediated degradation that happens at heterochromatin. And then CHP2 is working together with some of the deacetylases to prevent transcription from happening at heterochromatin. So transcriptional gene silencing, CHP2, post-transcriptional gene silencing, SWI6.
So I guess we have summarized your postdoctoral work very well, and then you started your own lab, obviously.
Yep.
So when this transition happened, what did you want it to work on in your own lab, and what were your goals when you started it?
Yeah, no, this is a really good question because, so I'll tell you, I was interested in this mechanism. And so let me start with the latest publication that we have, because we have the audience here. they have the sort of the pathway in their head. So one of the major sort of issues that bothered me for a long time, and I think it bothered the field for a long time, was in 2002 when people showed that RNAi is required for heterochromatin formation. If you looked at the chip results, when you knocked out Argonaut or when you knocked out the RNAi machinery, silencing was lost, but there was still a basal level of K9 methylation that persisted, right? So subsequent to that, many papers came out, including some from my lab and other people's lab, where they showed that small RNAs can nucleate heterochromatin at an ectopic site. So there was no question about the fact that small RNAs that are complementary to euchromatic part of the genome can recruit the entire heterochromatin machinery there. But at heterochromatin, going back, for example, to the centromeres, there was still this small K9 methylation. So everybody proposed in all their models, several, like 3 or 4 models that came out, everybody always placed Argonaut-associated small RNAs as the only mechanism for nucleating CLAR4, the histone-methyltransferase, at centromeres. But this wasn't satisfying to us. So we wanted to see if there was potentially other mechanisms that could be recruiting CLAR4 to this region. And I'll make a very long story short. Basically, through a lot of genetics and through a lot of really carefully designed experiments, we were able to identify a long non-coding RNA at the centromeres of fission yeast that can act as a silencer. So it has all the information to recruit CLAR4 all by itself. And at that, long non-coding RNAs, also 2 deacetylases converge. Remember, I was telling you about CHP2. Well, CHP2 interacts with this deacetylase called CLR3. And that also gets there by itself. And the other one is SIR2. So now you have the entire machinery there for deacetylating K9 and methylating K9. And this long non-coding RNA can do everything essentially by itself. And so this was really important because it told us that these long non-coding RNAs, there is a long non-coding RNA that can really work as a silencer. And this is kind of like the exist, RNA that exists on the inactive X chromosome. It's similar to that. It has all the information it requires in order for it to recruit all the factors. Now, why do we need RNAi? So I think the entire machinery is really tethered around this long non-coding RNA. And RNAi is kind of like the PCR machine. So what I mean by that is that, for example, in mammalian cells, nuclear RNAi really is not detected in somatic cells. So that is very controversial. The initial papers that came out, and essentially, we never worked on it, but essentially, people agree that this mechanism that we've discovered in fission yeast, even though it's prevalent and exists in the germ cells of mammalian cells and metazoans, it is not present in the somatic cells of mammals. But mammals have millions of copies of these long non-coding RNAs. Fission yeast only has 13 copies of this particular long non-coding RNA. So I think what has happened through evolution, again, we'll go back to evolution, is that fission yeast co-opted the RNAi machinery to amplify the long non-coding RNA signal by which it can recruit more CLAR4 and can establish heterochromatin at centromeres and other repetitive elements. By this long non-coding RNA is present at every single heterochromatic region in POMB. And we showed that in the absence of any K9 methylation or small RNAs, CLAR4 by itself can get recruited there. Okay, so I think basically what this is telling us is that the long non-coding RNA recruits CLAR4 and the machinery that's required for that through iterative cycles of deacetylation and K9 methylation. A basal level of K9 methylation can be established at heterochromatin independently of RNAi. Let's say RNAi is not there. Then RNAi comes in, amplifies the entire signal, and then you can get a full repressive state because K9 methylation, K9 trimethylation status goes up, and the recruitment of all the downstream factors like CHP2 and SY6 can now fully establish heterochromatin at centromeres.
So is this the only mechanism that would establish the H3K9 trimethylation-mediated heterochromatin?
At centromeres, these are the only two mechanisms, the RNAi and the long non-coding RNA. At the mating type region, there's also specific DNA sequences and also at telomeres. There are, in addition to what I just told you, RNAi and this long non-coding RNA, there are also specific DNA sequences that can recruit factors that are required for establishing heterochromatin independently of the machinery that I just talked about. Yeah.
Okay. So another thing you also worked on was the regulation of quiescence and how epigenetics, chromatin, and transcriptional changes regulate this process.
Exactly. That's a really good, So I think it's a really great story for people who are trainees and listening to this. This is a classic case where a mistake that you make in the lab pays dividends later in life. I was working on the CHP2 complex, in fact, and I was purifying the various components of this complex, and I was doing a very routine Western blot. So I had already worked with one of the proteins in that complex. I had purified it, I had characterized it, etc. And I just wanted to do a very routine Western blot. So I grew that strain that had the tag on it on that protein. But then I forgot to do the Western blot. So normally we harvest cells when they're in log phase, and then we do the Western blot. Well, I forgot about it because I was doing a bunch of other things in the lab. And I came in the next day and the cells were in stationary phase. I'm like, I'm going to do the Western blot anyway. So I did the Western blot and I could not detect the protein at all. It was gone. And so, all my positive controls were working. So I repeated the experiment, same thing. And then I said, okay, let's do a time course kind of a thing. And so I was able to show clearly that as soon as cells went into dormancy or went into stationary phase, let's call it here, the protein disappeared. So then I thought about it for the longest time and I wondered why. Why do cells, go through this process? Why do they get rid of this protein? And then I actually tested a bunch of other heterochromatin proteins and I realized that the expression of many of these genes changes dramatically. Some disappear, some their expression goes up, etc. So I wondered about what happens to constitutive heterochromatin when cells go into quiescence. So when I talked to Danish about this, Danish said, I'm not interested in stationary phase biology. When you start your own lab, you can work on this problem. So I said, okay, I think that's a good idea. I'll do that. So when I started my own lab, I started looking at the role of constitutive heterochromatin proteins. These are the proteins that I was working on in proliferative cells, now going into quiescence. And that's where we discovered something really profound. And that was that these proteins actually acquire a new function when cells go into quiescence. They are very important for establishing the transcriptional program of cells in dormancy. So when you put cells under long-term stress, the machinery gets co-opted to now turn off the expression of many genes throughout the genome, roughly around 700 genes in the euchromatic part of the genome, when cells go into quiescence. And so I think that was a really important discovery. The small RNAs are a part of it. Now we're going to be we're studying the role of long non-coding RNAs, obviously, in this process. But the most important thing that we're about to submit this paper very soon that came out of this work is that, okay, well, these proteins are conserved from yeast to human. What about in the human system? What happens there? And that's where we have a really nice paper. And finally, we got mouse data for this paper because the reviewers asked for mouse data, where we can show that there's a very specific resistant program that gets activated in cancer. And so we are able to show that these proteins are very important for establishing the epigenetic state of resistance program in many cancers. And by a transient inhibition of some of these enzymes, we're able to reverse resistance stably in a true epigenetic way. So only a transient exposure to the inhibitor is able to have long-term epigenetic effect where a cell state changes. So it changes from resistance to a sensitive. And now the sensitized cells can be amenable to treatment again. And so when we submitted the paper and we had figured out the mechanism, the reviewers wanted mouse data. So we spent the last 2.5 years collaborating with a bunch of labs and doing a lot of the work ourselves. And we are now ready to submit the paper fairly soon.
Yeah, that sounds like a like a marathon paper, right? You have the idea, you have the data ready, and then you have to do all those things to still publish it years later and working on it.
Exactly. Yeah, and that's the thing. Like, I mean, it was all the entire the impetus for me to go into quiescence. What came from the mistake I made in the lab, and I think that's the most important thing. If you make a mistake in the lab and it doesn't make sense, that's usually an interesting, and that's pretty much true with all the sort of like in my career. Like even in my PhD, it was kind of like that, where we made an observation and it just didn't make any sense. Why is this happening? And then that led to a really interesting biology that we discovered and published. So I think, my point is that when you get unexpected results, whether they're on purpose, whether they're just a routine control that doesn't work, or whether they are a mistake that you make in the lab, they always take you down a very interesting biological story.
Yeah. Another thing you published were some star protocols on shipping yeast. So what kind of optimizations did you propose, especially also for the spike-in?
Oh yeah, so so those are the couple of protocol papers that we recently published. Yeah, this is just really to help the community with the chip that they were doing. We were having some problems getting inconsistent results with chipping some of these chromatin binding proteins. And we read a bunch of papers, and this was something that was kind of emerging in a few papers. We realized that some people are using, so I'll talk about the first paper, the double cross-linking strategy. So double cross-linking strategy is really nice because it allows you to actually stabilize the interaction of the protein to chromatin. So, it all depends on the length, the arm length of the cross-linker. So formaldehyde is fairly short. So what we do is we basically cross-link with formaldehyde, and we also cross-link with a longer, cross-linker, a cross-linker that has a longer arm. And so that allows for proteins that were a little bit further away from chromatin to stably interact with histones, stabilize that interaction. When we do that, surprisingly, you actually get a lot less background and your peaks look better. And that was the part that was really surprising to us, that really nailed it for us. So that was the protocol that we published, and we use that on, we routinely use that now on all the chromatin binding proteins because I think it works so much better. It's something you need to determine empirically. Some chromatin binding proteins, they work fine with formaldehyde, so you don't need to use another cross-linker, but many of them don't. So that's where you need to use the second cross-linker.
So is this only true for yeast or also?
No, I think this applies to all organisms.
Okay.
So we're running those experiments right now in mammalian cells too. So that's the first. And then the second thing that we did is the spiking control. So the spiking control came because one of the reviewers for our cell paper, the one that Jasper published, asked for controls, additional spiking control for chip of a chromatin binding protein. Now, this had not done before. You obviously, you have spiking controls for chromatin marks like H3K9 methylation, K4 trimethylation, whatever. those are available commercially. We can buy it from you guys. You can buy it from other companies, and we can do that as a spiking control. But this reviewer wanted the spiking control for a chromatin binding protein. So we spent a lot of time thinking about this, and talked to a bunch of people and sort of collaborated and not collaborated, but brainstormed with a bunch of people. And finally, I was kind of like with Jasper and I, we were thinking about this and we realized, hey, you know what? What we can do is we can use a different species. So most of the chips that we're doing have a tag associated with the protein. So for example, we're doing flag IP of something, of a chromatin binding protein. So we realized what would be great to do is use Cerevisiae as a model. So in this case, what we do, we go into Cerevisiae, it has a flag tag on a chromatin binding protein. We do sort of like we purify that, we do the chip there, like we essentially create chromatin from there. And we use that as a spiking control for our POMBE experiments. So that's how we managed to solve that problem. Now, the fortunate thing was that we also decided, hey, this can be commercialized and can be licensed. So we decided to write a patent on it, and so we got the patent on that too. So we submitted that, and then now we're looking for maybe Active Motive or some other company that might be interested in licensing this from us, which would be great. So I think this is a really nice reagent for people to use in their experiments.
Yeah, that would be then also interesting for like mammalian work, right?
Precisely, yeah. It can be applied to any chip that anybody is doing as long as you match the tag to the tag that of the spike in, then you can do it in any system. So once we did that and used it as a spiking control, the reviewer was very happy with the data and accepted the paper. We were able to move forward with publication of the paper.
Perfect. We'll link to the paper in the show notes. So there's also currently a paper posted in bioRxiv where I did not find a peer-reviewed counterpart, where you investigate gene clustering and its influence on transcriptional coherence. Do you also want to talk about that, or is?
Yeah, I know. It was a really nice thing. The paper that we did in quiescence really was really exciting, because there were really three major things that came out of it. Two of which I've already talked about. One was the conservation. Is this possible that the same machinery is also really important for establishing the resistance program, for example, in cancer, in response to long-term persistent stress. So that's one bit of thing that we did. The second thing that we did, and just talked about the long non-coding RNA being a silencer, we found that in POMBE, in quiescence, in addition to small RNAs, there were regions in the chromosome where CLAR4 was getting recruited, but we couldn't detect any small RNAs coming from those genes. So that made us think that maybe there are long non-coding RNAs that can recruit CLAR4. And when we kind of came up with that hypothesis and we found some evidence to support that notion, we went back to POMBE and then identified the long non-coding RNA. So there was a third thing that came out of that paper that was totally unexpected. In the quiescence paper, we discovered that when cells go into quiescence, linear sections of the genome were becoming transcriptionally co-regulated by heterochromatin proteins. So we found 3 genes in one region, 7 genes in another region, 9 genes that were all neighboring in another region. And so we found all these clusters of genes that were becoming transcriptionally co-regulated throughout the chromosomes of POMBE. And when we looked at the genes that were present in these regions, we realized that they are in very divergent biological processes, so they weren't like thematically linked together. So then we thought about this for a while, but the repression is really important for cells to survive long-term in quiescence. So I thought about this for about 24 hours, and then I came up with the idea that genome organization is not random. What do I mean? So if you think about POMBE going into quiescence, you have 700 genes that need to be repressed in order for cells to survive. So instead of placing those 700 genes in random places in the genome, wouldn't it be more efficient if you put them in clusters next to one another? So even though they're from different biological processes, but having them in sort of next to each other would make it a lot more efficient to repress their expression when cells go into quiescence. So maybe this is the type of genome organization that's been under selection for a long time. And to think about it again, instead of developing 700 different ways to target 700 different genes in 700 different places in the genome, if you place them in, let's say, 70 places in the genome, then you have a much more efficient way of establishing that program. So then we said, okay, is this concept conserved in evolution? So we looked at species from yeast to human and we examined the genomes and we went for genomes that were highly annotated. The reason for that is because we wanted a lot of go biological processes to be present in these organisms. So we went with the best studied model organisms, Pombe, Drosophila, C. elegans, mouse, and human. And we asked, are there pairs of biological pathways whose genes tend to cluster next to one another versus random? And the answer was yes. So about 1,800 pairs of biological pathways we discovered that were that had this feature associated with them. And then when we look to see, are there similar biological pathways whose genes are next to one another, the answer was yes again. So we found that many of these higher Go terms whose genes were next to one another were very similar in all these different species. So we said, okay, that's great. We have this. So now the question is, but are these pathways actually correlated? So for that, we switched to human. So from here on, we did everything. We did the full, and this is purely a computational paper, by the way. We looked at it in human cells and we found that, yes, pathways that their genes tend to be next to one another are more transcriptionally correlated versus others. So when we looked at the genes so let me just make sure everybody understands this. So let's say you have one pathway that has 30 genes and another pathway that has 20 genes. It's not like all the genes are next to one another. A subset of those genes are next to one another. So for example, in pathway 1, you have 5 genes that are sitting next to 5 genes from pathway 2. Now, when we looked at the genes that were sort of in the same neighborhood, that were next to each other, we realized that many of them are transcription factors. So we said, maybe the more efficient way of doing this is put 2 transcription factors next to one another, and then it doesn't really matter where the genes are. So as long as the two transcription, and then you can co-regulate the entire thing. And anyway, make a long story short, this really allowed us now to identify thousands of previously undiscovered gene regulatory networks in eukaryotes. So that's the first paper. It's in bioRxiv. We've had a hard time publishing it because we're new into the evolutionary field. So we always get very difficult reviews back, and then editors don't trust us and everything. But anyway, so we're going to push that paper through over the next couple of months. Hopefully, I've been distracted with these other things, so I haven't been focusing on that.
So
we're going to push that through.
Can you see any correlation with TADs? I mean, that's the next obvious question,
absolutely.
Right?
That's one of the key questions that a lot of the reviewers asked. So because we're looking at genes that are next to one another, the probability for them to be in the same TAD is very high. So the way we asked that question was the following way. Remember, I was telling you about transcription factors being next to one another? Okay. So we asked, of all these in human, of all the transcription factors that are next to each other, how many of them are in the same TAD and how many of them are not in the same TAD? So majority of them were in the same TAD. But I said, okay, so the ones that are not in the same TAD, what happens to the correlation of the associated pathways? Are they still correlated or not? And the answer was a resounding no. So that's how we address that question. But, kind of like, yes, but they do, tend to be in the same TAD. Yes, that's absolutely the case. Yes. So we do have that together with that. So then now that we have all these gene regulatory networks, we ask what happens in cancer. So that's another paper that we're about to put on bioRxiv. And then so we looked at we looked at 15 different types of cancers. And we asked, can we use the establishment or loss of correlation among these gene regulatory networks that we identified as a prognosticator of cancer? And the answer was yes. We were able to accurately predict whether a sample was cancerous or normal with over 98% accuracy. In majority of the cancers, it was 99% accuracy. And then we said, okay, can we prognosticate the stage of the cancer? So we've specifically looked at prostate cancer because there's a lot of really nice clinical data. And because it's a very slow progressing cancer, we were able to put the high grade and the low grade cancers. And we used our platform to ask, can we predict whether something is high stage or low stage? And the answer was yes. So we were able to predict that again with 95% accuracy or something like that. I don't remember right now.
Mm-hmm.
But again, all this work was done computationally. And again, now that we have all these gene regulatory networks, we don't have to be limited to cancer. We can apply this concept to any disease state and ask whether, we can use this platform to to identify. But identification is not the only thing, is not the only application of this discovery. It can also be therapeutic. And let me explain why. So imagine that in a specific in normal tissue, this gene regulatory network is not established. But in cancer, it gets established. So by disrupting that, we may be able to identify new nodes of vulnerability in different kinds of cancers and completely develop a whole new class of drugs that nobody ever thought about before, just by looking at um, kinds of gene regulatory networks that get turned on in a disease state, and then they become essential for that disease state to persist.
So this would then also be, how would you disrupt those gene regulatory networks by inhibiting like one transcription factor and then it all falls apart?
Yeah, that would be the case, or both transcription factors, or maybe they're well-known pathways and they have their own inhibitors. So all of a sudden, you can use that strategy to apply that. But I think again, if you think about, and this goes back to my quantitative sort of upbringing, or at least my brain, that if you think about a cell as a closed system, which is very complex, if you disrupt a node of regulation in one part of the cell, it could have unpredictable consequences in other sort of parts of the biological processes that are working. And so that's the hope, is that we might be able to identify new nodes of vulnerability nobody even thought about before, because of these unpredicted interconnectedness of these biological processes throughout the cell.
Mm. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
For which topic? Are we talking about this one, or?
Your choice.
If I had a grant due tomorrow, what would I write in it today? What would be the topic of that?
Yes. I mean,
I think right now. Sorry, go ahead. Sorry, did you want to?
No, yeah, it's basically the question of what your lab will be doing in the next 5 years, right? So.
Okay. Yeah, so right now we're really focusing on so the gene regulatory networks that I just talked about. I think that's something I'm going to put aside for the time being. I'm really focused on the epigenetics, specifically using Pombe as a model, looking at quiescence and looking at the long non-coding RNA that we discovered and characterizing those things. And then the other piece is all this work that we're doing in cancer resistance. We are focusing primarily right now on renal cell carcinoma, even though we are also working on breast cancer. What I really hope to accomplish over the next 5 years is to develop a lot of the preclinical data required in order for us to move some of our discovery into clinic. I'm also talking to some investors, hoping to raise money so we can start our company, which primarily will be focusing on phenotypic plasticity. So for my grants, they're going to be targeting these two broad areas. One is overcoming resistance epigenetically in cancer setting, and the other one is fission-yeast recombinant biology and also quiescence biology.
Okay. So for the last 43 minutes, we've been on a journey through your scientific career. Did we miss something important, or would you like to add something?
No, I think we managed to cover most of it. I had a very unorthodox way of becoming a scientist. My life was full of up and downs and a lot of, I would say, dramatic experience, especially early on. I was born in Iran, and, I was alive during the Iranian Revolution and the ensuing war with Iraq. So I experienced the revolution and the war. So that was the sort of backdrop of my life. And then we left Iran in 1983, end of 83, beginning of 84, and we moved to Canada, and I grew up there. So I think that experience really shaped who I am and how I approach problems. And, I think it's increased my resilience. And then the other person that played a very central role in my life was my mother, who saw something in me, which I didn't see in myself at all. And she was the one who always pushed me towards science and making sure that I sort of used she always told me, you have a gift and you need to use it. And if you don't do it, then it's a waste. So she was the one who really was the architect of my life in many ways, and pushed me towards science and everything. And I'm very happy that she did, because clearly it's fun. Being a scientist is a lot of fun, and in the process, if I can contribute to the betterment of humanity, why not? It's what life is all about.
So that's a good point to end this interview. I will skip my last question, and thank you, Mo, for being part of the show.
Okay, yeah, thank you so much.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.